Now, when a thief broke into a car, he cut his arm on the broken glass. Okay? Scientists, these bright guys, extracted DNA from the blood found in the broken glass. Now, you must remember, this is also a very small sample. But they only need a little sample to find basic DNA. Okay, now, they analyzed the DNA sample and prepared the DNA from 3 suspects, P, Q, and R. All right. The table below shows the results of the analysis of the DNA for each source. Now, people, you are going to get something like this in any one of your tests or exams. Remember that these things here, okay, are called bands. And those bands are produced when they use electrothesis and plating. All right, so that it's an incredible process that they use, but this is so accurate. Now, something I need to tell you, in our human genome, which is the way our DNA and everything our DNA is made up of, human beings, okay, walking on 2 legs, us, you, me, everybody, any color, it doesn't matter what color, it doesn't matter whether they're big, small, tiny, we all have the same human genome. All right, and there's something like 3.2 billion. It's almost like the money that gets wasted on so much nonsense here, but 3.2 billion base pairs, okay, that make us who we are. And only 1% is different. So if you look at the person next to you, or tomorrow when you're at school, look at the people around you in your classroom. What makes each one of you individual and absolutely unique is 1% difference in your genetic makeup. 1%. Do you know how little that is? And that will account for all the differences that you see between human beings. So we are all the same. And we should all be looking after each other, but that's just another little snippet of useless information. So here we go. All right, let's look at our broken glass and our everything here. Okay, first thing you're going to do is you're going to line up your bands. So this band here for the broken glass lines up with Q. And this one here lines up with P, and it lines up perfectly with Q. And this is how you're going to work it out in a school. You're going to take, or in an exam, you're going to take, okay, this one and this one is going to line up with this one and sort of with that one. This one lines up perfectly there. It lines up perfectly there, but also with P, and it lines up perfectly there. What does this tell you? That's the sample that they found. It means that it is Q's blood, irrefutably. But what you can also see here is that because P and Q are quite similar on 3 spots, they could be brothers. Okay, and the fact that the police found all 3 of them means they're probably friends, but yeah, we won't go there. We're not playing CSI now. So what do the diagrams above represent? Well, that's easy. It's a DNA profile. Okay, it's the DNA profile. Then, which suspect is most likely the thief? Well, Q and the broken glass sample are 100% match. So you just say number Q. Okay, there we go. Give a reason for your answer. Well, the reason is easy. Q's bands are identical to the sample of blood found. Okay, so it's as easy as that. It is identical. There is absolutely no argument there that it is his blood that's on that glass. How the blood got there, well, that's for the courts to now sort out. Now it says name 2 possible disadvantages using the evidence in a court of law. Well, we've just said this. Well, number 1 is going to be human error. And that human error is in the collecting and the processing, analyzing, let's say processing of the samples. You know how when you watch movies or you watch CSI, whatever, And they, I mean, the Oscar Pistorius's murder trial that everyone watched in South Africa quite religiously, okay, it was contamination of the proof, of the evidence, and everyone, contamination, they didn't wear slippers, they didn't do this, they didn't, so it's human error. Big issue, human error. Okay, then second thing that's also a problem is there's only a small amount of DNA. Only small amount of DNA. Okay, next one is you can have planting, I don't know how else to put this, planting of false evidence. So I can go and I can take, for example, my producer Cheryl, and I can get a little bit of her blood because maybe she cuts herself on a paper, and I then go and I put her blood somewhere, and she's then responsible for murdering somebody when she never did anything. She wasn't even there. Okay, so planting of false evidence. And our next one is that there could be an identical twin. Suspect could have identical twin. Okay, so any two of those. Right, next one. Provide two benefits of DNA profiling other than solving crimes, or people, really. Number one is paternity tests. Okay, a blood test can tell you who is not the father, but a blood test can't tell you who is the father. So let's say the baby has AB blood, the mother is O, okay, or hang on, the mother is B. The father either has to be A, O, or A, that's it. Okay, now you can prove that it'll say, okay, they're not the parent. But if you've got two fathers and they've both got the same blood types, because remember, there are only 6 blood types, and then you've got your positives and your negatives, you need a paternity test. Then number 2 is for genetic diseases. So to check if people have genetic diseases like Huntington's chorea. 3, to identify a dead body. Identify a dead body. Okay, the body's there. There's no identification on it. With a bit of luck, there's DNA somewhere and you can find it. Okay, then you've got, I'm running out of time here, to research the animal populations, okay, and variations in those animal populations and species. They also use it to track an individual in a population. For example, I know they do it with cycads, with really expensive dogs and really expensive cats. There's somewhere that, because they've got very specific gene markers, they can pick it up. Alrighty, next question, and I think that was the last question. Explain one precaution you would should be taken when working with DNA samples. Well, the most important one would be careful handling to prevent contamination. And just off the top of my head, the next important one would be very careful marking. So marking and labeling and storing. Okay, because if it doesn't help getting the stuff there, the information, and then what happens? Well, the proof is gone. Okay, you can't have that. Alrighty, let me just see. Next question. Here we've got, this was a multi-choice if I remember correctly. So we've got a person was seriously injured during a fight. Samples of the blood were taken from the injured man, okay, and the crime scene. These samples are compared with blood samples, blah, blah, blah. So we've got 1 to 4. So we're going to have a look here. There's our victim. There's the crime scene. So again, you look at your lines. So sample, nothing matches. Mind you, this one matches. All right, this one, nothing matches. This one, nothing matches. This one, that one matches. Nothing, nothing, nothing. And I think we have a problem here. That one matches. Oh, mind you, this one matches too. So your suspect is number 3. We've got one more question with this stuff, and we're finished. This diagram shows the DNA profile of a child and her mother and 4 males. So they're trying to figure out who the daddy is. So this is, there they tell you, is paternity, to establish DNA paternity. So let's see here. If we look here, we've got the child, the mother, the male. Okay, so we're not worried about the mother. we're looking at the child. So what corresponds? Male 1 corresponds, male 2 corresponds. Okay, then here, male 1 and male 3 corresponds. And then we look here, child and again male 3, and child and again male 3. So male 3 is the daddy. All right.